Bioinfocongress 3. Doçent Doktor Derya Sargın, sosyal davranışın altında yatan sinir devreleri. Doçent Doktor Derya Sargın, lisans derecesini Boğaziçi Üniversitesi Moleküler Biyoloji ve Genetik alanında almış ve Almanya Göttingen'deki Uluslararası Max Planck Araştırma Okulu'nun nörobilim alanında yüksek lisans programını tamamlamıştır. Max Planck Deneysel Tıp Enstitüsü'nde Dr. Hannelore Ingenreich ile yaptığı doktora çalışması, nöropsikiyatrik hastalıkların patofizyolojisine katkıda bulunan moleküler ve hücresel mekanizmaları aydınlatarak, şizofreni ve bipolar hastalığı olan hastalarda yeni ve etkili tedavi stratejilerinin geliştirilmesine imkan sağlamıştır. Doktora sonrası ilk çalışmasını Hasta Çocuklar Hastanesi'nde Dr. Sheena Jocelyn ile birlikte duygusal hafıza oluşumu ve tahsisi üzerine tamamlamıştır. Daha sonra Dr. Evelyn Lamb'in Toronto Üniversitesi'ndeki laboratuvarına geçmiş ve burada serotonerjik transmisyonda yeni kronik stres kaynaklı düzenleyici mekanizmaları belirlemiştir. Hotchkiss Beyin Enstitüsü ve Alberta Çocuk Hastanesi Araştırma Enstitüsü üyeliğine sahip olan Doçent Doktor Derya Sargın, 2019 yılı Temmuz ayından beri Calgary Üniversitesi Psikoloji bölümündeki laboratuvarında çalışmalarını sürdürmektedir.
Bioinfocongress 3. Associated Professor Dr. Derya Sargın. Neural circuits underlying social behavior. Associated Professor Dr. Derya Sargın received her bachelor in molecular biology and genetics from Boğaziçi University, Istanbul, Turkey, and attended the master's program in neuroscience of the International Max Planck Research School in Göttingen, Germany. In her doctoral research with Dr. Hannelore Ingenreich at the Max Planck Institute for Experimental Medicine, she identified molecular and cellular mechanisms that contribute to the pathophysiology of neuropsychiatric disorders and led to the development of new and effective treatment strategies for patients with schizophrenia and bipolar disorder. She completed her first postdoctoral studies on emotional memory formation and allocation with Dr. Sheena Jocelyn at the Hospital for Sick Children. She later moved to the Dr. Evelyn Lamb's laboratory at the University of Toronto, where she identified novel chronic stress-induced regulatory mechanisms in serotonergic neurotransmission. She has been working in her laboratory at the University of Calgary Department of Psychology since July 2019, and she's a member of the Hotchkiss Brain Institute and the Alberta Children's Hospital Research Institute.
Sayın Doktor Derya Sargın ve değerli katılımcılarımız, Bioinfocongress 3 etkinliğimize hepiniz hoş geldiniz. Değerli hocamız bizlere bugün Neural Circuits Underlying Social Behavior konulu bir konuşma gerçekleştirecektir. Konuşma bitiminde ekibimizin hazırladığı ve katılımcılarımızdan gelen sorulardan oluşan soru-cevap bölümümüz olacaktır. Merhaba Derya Hocam, tekrardan hoş geldiniz.
Merhabalar. Öncelikle davetiniz için çok teşekkür ederim.
Biz teşekkür ederiz hocam.
Çok güzel bir event ortaya koymuşsunuz. Bu kadar değerli bilim insanı arasında yer almak gurur verici. Teşekkür ederim.
Teşekkürler hocam. Dilerseniz sunumunuza başlayabilirsiniz hocam. Ekranınız görünüyor şu an.
Tamam. Sunumu İngilizce olarak yapacağım. Ama soruları Türkçe sorabilirsiniz. Okay, so in my lab, we are looking at the mechanisms of how specific neural systems encode behavior. We particularly focus on emotional and social behaviors and how early insults such as stress has an effect on neural function. So one line of research that we are pursuing at the moment is understanding how hypocretin orexin systems, and hypocretin orexin, I use these interchangeably, they are how these systems are involved in social behavior and how social isolation stress affects the activity of these systems and their downstream pathways. Today, I will present some of our new data focusing on orexin, hypocretin, and social behavior. The social behaviors are evolutionarily conserved and exist in a diverse group of mammals, ranging from rodents to humans. They are very important for our communication, also for our interaction, reproduction. So they are very critical for the survival of our species. Social interaction involves detection of cues in the environment, cues to various sensory modalities, and then responding to these by determining the appropriate action. So in simple terms, acute sensory information from one animal, one conspecific, is detected by the other one and transformed into a behavioral decision. So we have to keep in mind that social behaviors are quite complex. They occur at a fast temporal scale, and no doubt they involve multiple circuits that interact with non-social cues in the environment, and this leads ultimately to that decision-making of the animal. So many things are important in this. Animals' internal states, past experiences, all of these things are very important in this reciprocal behavioral interaction, which makes social behaviors among the most complicated type of behaviors. We have ample evidence that suggests various hypothalamic subregions to be important for these socially relevant internal states. Hypothalamus, as you may know, is an evolutionarily conserved brain region. It's very important for homeostatic regulation. And there are quite a few regions that are implicated in social behavior, subregions of the hypothalamus. So you can see here that the medial preoptic area, for example, it regulates sexual behaviors. Neurons in this region, anteroventricular periventricular nucleus is critical for maternal bonding with pups. Ventral hypothalamus, ventromedial hypothalamus is very important in aggression and mating. So the common underlying process for all of these social behaviors is detection of the social cue, which leads to arousal, attention, and followed by motivation, all together contributing to the ultimate decision-making for the behavioral output. And these arousal, attentional, motivational processes, these are also functionally shared by another region of the hypothalamus, lateral hypothalamus. This is a key region in the regulation of many different processes. So these range from sleep, arousal, feeding, motivated behaviors, reward-related behaviors, energy balance, homeostasis, and stress. So the cell population in the lateral hypothalamus that has been mostly studied with respect to these functions are called hypocretin neurons or orexin neurons. Hypocretin and orexin are basically used interchangeably. So these neurons form the arousal hub in the brain. They are located here deep in the brain. Their absence leads to narcolepsy in both humans and animals. And the term arousal here indicates not only wakefulness. So I want to take your attention here that multiple different acute stressors can actually increase hypocretin activity, while impairing hypocretin function can just do the opposite. So as all of these modalities hypocretin regulates are critical for social behaviors, we asked whether hypocretin plays a role in the regulation of such behaviors. So we have, there are a few important studies that actually indicates this might be the case. So these studies have been performed in the Sakurai lab. So Sakurai lab together with they are from Japan, and together with the Lucera lab from Stanford, they were the first to identify the presence of these hypocretin neurons in the brain. So they showed that, so they characterized orexin receptor knockout mice. And what you see here is a task that is called 3-chamber social interaction task to detect sociability in mice. So here there is an empty compartment, and the other compartment has a mouse, a stranger mouse that is completely new to the experimental animal here. It's confined in a pencil cup like this. And mice are quite social animals, curious. So rather than spending time in this empty environment, they want to interact with this animal here. As you can see this in the wild type animals, that they spend actually more time interacting with in the social zone. But if you look at the orexin receptor knockouts, we don't see this. So they do spend equal time, whether empty or the social zone. So this means that their sociability is actually reduced. We do have some data from orexin knockouts as well, from a study that has been now performed some years ago, but they showed that in a resident intruder test that, again, all the behavior responses to the stranger animal is reduced when orexin is knocked out in the brain. So we also have some evidence from human studies. So these patients, these are epileptic patients, and what you see here is a microdialysis probe that is inserted deep in the brain. So they basically collected dialysates from the brain of these patients and assayed for peptides. And one of these peptides were hypocretin. So what they found is that hypocretin levels were maximum when patients are engaged in social interactions, as opposed to all the other things that they do throughout the course of the day. So these studies suggested that the orexin or hypocretin may be involved in social behavior. Today, I will present you some of the new data from my lab that shows that with social interaction, hypocretin neurons are robustly activated. And social interaction-induced activity in these neurons changes based on the nature of the social encounter, meaning that friend or foe, the activity differs. And we could show that the hypocretin activity is actually necessary for certain types of social interaction, but in a sex-dependent fashion. So in my lab, in order to observe neural activity in behaving animals, we use a technique called fiber photometry. So to see how neurons are engaged in social interaction, we measure hypocretin neuron activity while the mice are running through certain tasks. To do this, first we inject viruses, AAV viruses, encoding calcium sensors into a specific brain region. So when neurons are activated, calcium is released, and this released calcium binds to the sensors, increases GFP fluorescence, and by increasing this fluorescence, we can visualize the neural activity. Implantation of an optical fiber into the specific brain region of interest allows us to deliver the excitation light and collect overall fluorescence. So we are using a hypocretin-Cre knocking line that has been developed and kind of provided to us by our collaborator, Dr. Gina Leininger from Michigan State University. And all of this work now I will present have been conducted by my PhD student, Matt Dawson, and who got technical help with surgeries from our technician, Naila Jamani. So we infuse Cre-dependent AAV viruses encoding the calcium sensor GCAMP into the lateral hypothalamic region in this hypocretin-Cre mice. So this allows us to express GCAMP selectively in hypocretin neurons, as you can see here. And then we implant the optical fiber right above the lateral hypothalamus, and we record the activity of orexin neuron populations during behavior. So we first use one of the conventional social interaction paradigms that tests sociability in mice, which I have mentioned earlier, 3-chamber social interaction task. So this task does not allow direct social interaction. So I want to take your attention here that they do not directly interact with each other because this animal is confined, right? Stranger is confined in a pencil cup like this in one corner of the chamber, and the other one is kept empty. So mice are socially curious, so they spend actually more time investigating here in the social zone rather than this empty compartment. So using fiber photometry, in vivo calcium imaging, we examine the activity of hypocretin neuron populations as this animal goes between the two different zones and sniffs the stranger versus this empty cup. So what did we find? Here you see the example traces, hypocretin neural traces from a female and a male mouse. And you can see the activity as the animal goes between the social sniffing zone and the non-social, this empty sniffing zone. So we quantified the amplitude and the frequency of hypocretin activity peaks when the mice are going between these different zones. So we could see that in red here, you see the activity when mice are in the social zone. When mice are sniffing the stranger, we could see that the amplitude of hypocretin activity, hypocretin calcium responses, and the frequency of, again, hypocretin activity peaks seem to be much larger compared to when the animals are in the non-social zones. And we could see this in both females and males. Here, what you see is the area under the curve of the hypocretin calcium responses, again, when the animals are interacting with the stranger versus the empty object. And again, we could see that this time, which is more strongly presented in males, that all the calcium responses aligned together seem to be much greater when the animals are in the social zone. So overall, we could see that the hypocretin activity is greater when mice are interacting with a stranger as opposed to sniffing an object, such as an empty cup. Now, the three-chamber task, it prevents direct social interaction, which avoids aggression, but it doesn't actually allow for natural interactions to occur. So we next performed fiber photometry when the mice are freely interacting in their home cage. We performed these experiments first in mice living together in a cage. So they know each other, they are litter mates, they have been together all their lives. What we do in this task, here you see the one with the fiber is the experimental animal. So we separate the experimental animal from its cage partner for about 10 to 15 minutes. We first record the baseline hypocretin activity in this experimental animal when the mouse is alone in the home cage. And then we introduce the familiar animal, the cage partner, back in the cage, and record the activity of hypocretin neurons while mice are interacting. So what did we see? This interaction, so in gray lines here, you see the interaction, social interaction. When the mice are interacting, we could see that as soon as the social interaction starts, there's a pronounced increase in hypocretin activity. So this is right at the time when mice are interacting. And social interaction is quite robust at the beginning. After this peak in activity, you can see that the hypocretin calcium response goes down, habituates over time. It's quite high when the mice keep interacting for the first couple of minutes, but it gradually habituates and declines as the social interaction also decreases over time. Now I will show you what happens in real time. So on the left here, you will see the mice interacting with each other. On the right, you will see the hypocretin activity in real time. So this is our experimental animal tethered to the optic fiber cable here. We put the familiar mouse in, the cage mate. They interact. As they interact, you can see that the hypocretin activity increases. And the interaction, as you can see here, is quite robust at the beginning. So as this interaction goes on, hypocretin activity also stays quite high. Now, these are mice that know each other, right? They are familiar cage mates. But what happens when we introduce a mice who are completely stranger to each other, that they have never met before? So we introduced next this novel animal, age and sex matched, into the cage of our experimental animal. again, it realizes the other mouse interacts. As it interacts, the hypocretin neural activity increases. But notice that this increase is now much larger. And again, interaction is robust at the beginning, so activity stays high. So what did this show us? Okay. So I'm going on. I'm very sorry. Something happened to my computer and it just turned off. Anyway, so starting again, here I showed you that basically what we could find was that the interaction between familiar and unfamiliar animals was differentially encoded by orexin neurons. This is what we could see. So we quantified the calcium signals, hypocretin calcium signals over time in females and males when they are interacting with these different animals, familiar and stranger mice. And what we found was that, you can see here, so these are average calcium responses over time. So in response to, and they're all aligned to initial approach, social approach. So in response to that first approach, we could see that the activity becomes really maximum. And over time, they start habituating. So the hypocretin activity habituates and declines. And again, we could see that in both females and males, we could see a greater activity when animals are interacting with strangers. So we quantified this. Here you see the area under the curve of the calcium response in both females and males. And here's the maximum hypocretin activity. So when animals are, again, interacting with the stranger mice, we could see that this activity, hypocretin activity, is significantly higher in both animals. But males overall seem to show increased hypocretin activity during social interaction. Okay, so to be able to understand this further, we wanted to look at the interaction and activity over time. So how does this change over time? So here again, we see that maximum activity that happens when the animals meet for the first time is at the beginning. And this is what you show here is hypocretin activity over time in females and males. And in relation to this is the behavior, social interaction over time. We see a very similar trend with hypocretin activity and behavior. And the first interaction is quite high at the beginning, and it also declines over time. And overall, what we see is, again, males, the hypocretin activity in males and social interaction in males seems to be much higher than the females. Okay, so moving on, next we asked how hypocretin activity will change if the experimental animal is in a novel environment, novel cage. So not in its territorial and familiar home cage anymore, but in a novel environment in which the context would be completely new for the mouse. So what happens in a novel setting first? Behaviorally, mice spend more time engaging in different kinds of non-social behaviors like exploration, rearing, ambulation. So they spend more time in this as expected in a novel environment. But we also determined that the time that mice spent interacting with strangers or familiars seemed to be unchanged in the novel environment. So in line with this, what we could see, similar to the home cage, we could see that interaction-induced hypocretin calcium responses were much higher. They reach maximum levels right after the initial social approach and decline to baseline over time in both sexes. And this is, again, the quantification. So we see exactly the same thing in the novel environment. And with males, again, male activity being overall higher than the females. So this shows that whether it's a home cage or a novel environment, we could still see the same activity levels. So we wondered if this differential response that we see will present itself in response to any new situation, any novel situation. So is this generalized or is it specific to social interaction? To test this, we performed fiber photometry when mice are interacting with interacting with novel or familiar objects in their home cage. So the mice were made familiar with the objects by being exposed to these objects for a few days in their home cage. Then these objects were presented to the mouse in its home cage while we are performing fiber photometry. So again, these are average calcium signals over time. In both females and males, we could see that hyperkeratin activity increases with the initial approach and interaction with objects. But what we don't see is that differential response. So the maximum, when we quantify the maximum hyperkeratin signal, we cannot see that it differs whether the object is new or familiar to the mouse, and actually increases much less. So then we could see that the significant difference that we see in social interaction is absent. This differential activity seems to be more specific to social. So one important and interesting finding in our study was this. So the relation between this initial maximum hyperkeratin activity and subsequent behavior. In both home cage and novel cage conditions, we could see that there was a significant correlation between the initial, that high maximum hyperkeratin activity, and the subsequent duration of social interaction. So this initial large signal initiated by the first interaction predicts how much stranger mice will interact with each other in the subsequent minutes of the task. So having established this link between hyperkeratin activity and social interaction, we next wanted to see how necessary these neurons are for social behavior. So these experiments have been performed again by my PhD student, Matt Dawson, and our star technician, Mio Tsutsushi. She's an excellent surgeon. So we infused Cre-dependent AAV viruses encoding a light-activated inhibitory channel, chloride channel, into the lateral hypothalamus. So this allowed us to express this inhibitory channel specifically in hyperkeratin neurons, as you can see here. So because of the importance of that initial hyperkeratin signal that determines the subsequent interaction, we photoinhibited hyperkeratin neurons prior to when mice meet for the first time, and we kept the light on during social behavior. And we introduced familiar and stranger animals in a counterbalance fashion. So mice, we could see that in males, mice spent significantly less time interacting with the conspecifics when the hyperkeratin neurons were inhibited. Here you see in green the ones that were infused with IC, where inhibition occurred, and EYFP, the control animals, are in magenta here. So photoinhibition caused a deficit in the social interaction of males, but if you look at females, we don't see this. So in contrast here, optogenetic inhibition of hyperkeratin neurons in females did not affect social interaction. So we have a sex difference there. So we repeated this photoinhibition in a more controlled setting in the 3-chamber social interaction task, where the novel mouse was rather confined. And you can see here is the activity of mice in the chamber. So similar to the home cage environment, resident intruder test, we could see that male mice, normally the controls spend more time interacting with the social animal, but here, when hyperkeratin neurons are photoinhibited, sociability is gone. Sociability is reduced, but we don't see the same effect in females. Deficits in hyperkeratin function was previously associated with an inability to maintain a wakeful state in humans and rodents. So any change in arousal, we thought, may contribute to these behavioral differences that we're seeing by hyperkeratin activity manipulation. And to test this, we determined locomotor activity during photoinhibition of hyperkeratin neurons. We performed open field and elevated plasmase tests and looked at how much distance mice travel when their hyperkeratin neurons are inhibited. In either test, females and males, we did not see any difference in the locomotor activity upon acute hyperkeratin inhibition. So we went on to see, again, because of the potential involvement of these neurons, of hyperkeratin neurons, in stress response, we thought manipulation of hypocretin activity may also interfere with the generalized anxiety states, which may actually be the reason we're seeing some social differences. But again, in open field and elevated plasmase tests, so these tests are done to be able to look into anxiety-like behavior in mice. So if they spend more time in the periphery here, and if they spend more time in the closed arms, this indicates a greater anxiety-like behavior. So we assessed the time that they spent in different zones, but either the animals were infused with the control EYFB virus or the IC, inhibitory channel, we did not see any changes in females and males with acute inhibition of hypocretin neurons. This showed us that the general anxiety was not indeed affected with acute hypocretin inhibition. So to sum up this part of my talk, we showed that hypocretin neurons exhibit this robust increase in activity in response to social interaction, with activity levels larger in males. And there's a discrimination between unfamiliar and familiar individuals, and this is differentially encoded by hypocretin neuron activity, as it's much larger when animals are interacting with novel mice. This initial large increase seems to be determining the subsequent duration of behavior. And signal during interaction with familiar or novel objects does not show this differential activity that we saw with social interaction. So this showed that the differential activity that we observe in hypocretin neurons is not a response to general novelty. And acute optogenetic inhibition of these neurons disrupted social interaction in males only, without any effect in females. So this showed a sex dimorphic effect in the necessity of hypocretin neurons for social behavior. Now I'm going to change the gears a bit, and I will show you how social isolation stress affects the hypocretin activity and social behaviors. So the last couple of years made us think about isolation. We felt isolated more than ever. We had to be socially distanced to stay healthy, and looking forward, it felt more like a marathon rather than a transient period. So we felt somewhat isolated, tired, worn out. We don't really know the long-term impact of this. So we may have been experiencing isolation more, but it's not a new phenomenon. The risks associated with social isolation have long been studied. And we know that isolation has a significant impact on our physiological and mental health. It's actually associated with a higher likelihood of mortality at a rate of smoking 15 cigarettes a day or having an alcohol use disorder. And it seems to be twice harmful to our health compared to diseases like obesity. Reduces lifespan for all genders and races studied. And among the cause of death, suicide seems to be the most common, as social isolation increases the risk for mood disorders. So we see that certain age populations are affected more. Aging population, children, adolescents, especially the adolescents and teenage population, I mean, they are so social driven that isolation has significant effects on them. So within the younger population, we could see that those who experienced enforced isolation are 5 times more likely to require mental health service. There are also long-term effects. There's association with the generalized anxiety or social anxiety. Altogether, loneliness in kids results in future mental health problems that could be detected up to 9 years later. So we do have abundance of information that comes from children reared in institutions all over the world. Recently, the number of children living in institutions reported to be 8 million, although many remain unregistered. Years of research showed impact of social deprivation on brain development and long-term mental health. And we could see that the deprivation, social deprivation, especially early in life, is harmful, which shows us the importance of a critical period. So there's one major question. What are the changes in the brain? What are the mechanisms that are underlying these long-term changes? So in my lab, one of the main goals is for us to understand how isolation during a critical developmental time window leads to long-term social and emotional deficits. We think that disrupted circuitry underlies these behavior dysregulation, and our aim is to come up with treatment approaches that can act at the level of circuits to treat long-term deficits that arise from childhood adversities. So to do this, to look into the mechanisms, we're using a mouse model of chronic social isolation. It's a validated and widely used model of isolation rearing. At postnatal day 21, this is also the time that mouse pups are weaned from their moms. So at this time, we single house half of the litter mates and until adulthood, and controls are kept together in the cage. So socialization is very important for humans, but we see this in mice as well. Chronic social isolation interferes with their socio-emotional development because they are deprived of these critical social interactions, social play, that normally helps guide establishment of their adult social behavior. So just like children, using this model, my earlier research focused on the disturbances in the serotonin system in response to social isolation. Serotonin-producing neurons, they are located deep in the brainstem here in raphe nucleus. They have important roles in regulation of social and emotional behaviors. So we have several publications showing the deficiency in the overall activity of serotonin neurons, both in the raphe and in downstream projection regions, and some of the physiological mechanistic changes underlying these. But I won't go into detail today. But we, along with many others, identified the long-term emotional deficits with this post-weaning social isolation in mice. But one particular finding is the basis of what I'm doing in my lab today. So serotonin neurons, they receive numerous modulatory inputs. There are dense excitatory inputs from hypocretin neurons onto serotonin neurons, as you can see here. So, well, we could see that these are patch clamp recordings from slices obtained from either a control animal or an isolated animal. Well, we could see that hypocretin normally excites serotonin neurons, as you could see here, the increased frequency of action potentials. But we see in isolated animals, the orexin-induced response of serotonergic neurons seems to be much reduced. So there's an interaction between these two systems, hypocretin and serotonin systems, that seem to be disrupted by social isolation. So based on our earlier work, we hypothesized that hypocretin and serotonin circuits are central components of a larger network that may be disrupted by social isolation. And we asked the following questions. How does chronic social isolation impact long-term social behavior? How is hypocretin neuron activity affected by social isolation stress? And what would be the circuits involved in this? Now that we established orexin hypocretin is important for social interaction. Now, how is this system affected in response to social deprivation? This second part of my talk is focused on the long-term effects of social isolation. And I will show you that isolation leads to long-term deficits in normative social behavior. Social isolation disrupts hypocretin neuron activity. And finally, I will show you some of our very recent data that suggests the role of the lateral hypothalamus dorsal pathway in isolation-induced deficits. Now, depriving rodents of social interaction during a critical developmental time window, this is post-weaning until adulthood. This is utilized as a model of early life social deprivation. We and others show that isolating mice leads to long-lasting behavioral deficits in adulthood. However, less is understood about how isolation actually leads to produces deficits in social behavior. So we did this. We looked at differences in social behavior in isolated and controlled mice. We could see that isolated mice in adulthood spent significantly less time interacting with novel conspecifics, with strangers in their home cage or in a novel environment. So we could see decreased sociability in isolated animals using different tasks. Next, we wanted to determine the activity states of the hypocretin neuron populations in controlled and isolated mice during social interaction. Now, these mice don't have a partner. So we measured activity in response to interaction with a stranger novel animal in their home cage. So to automate quantification of social interaction, we actually use a machine learning based open source analysis. So we track the mice, multiple mice, using deep lab cut, and then we analyze their behavior using Simba. So the numbers you see here just shows that different types of sniffing as the mice are interacting here. So on the left, again, you will see the mice interacting. On the right, hypocretin neural activity during this time. So the resident here is a control animal. So let's see, they interact. And as they interact, as we've seen before, hypocretin neural activity increases. And this interaction goes on at the beginning quite a while. And then activity is higher during this period. Now you will see what happens with an isolated animal. So in pink here, this is the isolated mouse. It's a little bit hesitant approaching. So, but it approaches and interacts, and then goes back and interacts again. And as you can see, what we realize that the hypocretin neural activity is reduced during this interaction when the mice were isolated. So we quantified this. So we could see that the calcium response of hypocretin neurons. So first, similar to our earlier observations, in controls, we do see an increased signal as the mice approach each other and starts interacting. But if you look at isolated animals in red here, their hypocretin activity seems to be much reduced, much blunted after chronic social isolation. So going back to the slide that I showed you at the beginning. Now, we have some evidence that hypocretin activity disrupted after social isolation. We also have evidence that hypocretin-induced activity of dorsal-raphe serotonin neurons disrupted in social isolated mice. We have substantial evidence that hypocretin neurons from the lateral hypothalamus synapse onto dorsal-raphe neurons. They actually send direct inputs, majority of them being within the serotonin neuron proximity. So this pathway contains one of the densest hypocretin projections in the brain. What does orexin do? Orexin really activates the majority of dorsal-raphe serotonin neurons, and this activity is actually synchronized as shown in sleep and waking states. But how is this pathway involved in social behavior, and what happens to this pathway in response to social isolation? Dorsal-raphe neurons, serotonin neurons, they have been implicated in social behavior, and this has been shown in several different ways. Absence of serotonin in the brain impairs social interaction. There's a study published now some years ago showing fiber photometry during interaction between experimental and intruder male mice. So you could see here the GCAM fluorescence in serotonin neurons peaks, not actually similar to what we see with the hypocretin neurons when mice meet for the first time. And when they tested this with objects, again, similar to what we see, you see an increase in serotonin activity, but not as much. So social interaction seems to activate dorsal-raphe neurons more effectively than general novelty. And we tested social interaction and serotonin neuron activity in our mice that express GCAM selectively in serotonin neurons. So we inserted this time optic fiber just above the dorsal-raphe. So what we observed was that, as you can see here, when the experimental mice interacts with the intruders, serotonin neuron activity increases. As you know, we were seeing with hypocretin neurons, that serotonin neurons are responsive to social interaction. So what about the pathway? What about the response of hypocretin neurons that project to dorsal-raphe? To see how this pathway is involved in social behavior and isolation, we first injected the dorsal-raphe of the hypocretin Cre mice with Cre-dependent retrograde GCAMP virus. So after 2-3 weeks, the retrograde transport to LH is complete, and we implanted the optic fiber above the lateral hypothalamus to record from dorsal projecting orexin neurons during social interaction. So these are very preliminary results, and they have not been published. All are unpublished here. So in control mice, as you can see here, we can see clear we can see that the orexin projection neuron activity increases in response to social interaction. But in isolated animals, what we see is that this dorsal projecting hypocretin neurons seem to be much reduced in activity. So this gave us an indication that this pathway involved in social behavior and is disrupted in response to isolation. So to sum up my talk, we show that isolated mice show social deficits in adults. So they have disrupted social interaction. They spend less time interacting with strangers. They interact less, however, when they do engage in interaction, we can see that the hypocretin activity is blunted. And our earlier and now new data suggests that the hypocretin dorsal pathway may be a part of a larger circuit that is involved in this isolation-induced disruption in hypocretin neuron activity. So where do we go from here? We're wondering how does social interaction affect the activity of orexin projections, right? Hypocretin projections. So what we're doing right now, perfused mice 90 minutes after social interaction, which allows us to visualize CFOS expression all over the brain. So the CFOS is a proxy for neural activity. So we're looking into particular regions, performing brain-wide imaging and atlas registration of the activated neurons or those that show us differential activity patterns. So we are doing this together in collaboration with the App Lab, who developed this technique. So this allows us to see how social interaction affects the circuits and how hypocretin neuron inhibition affects the circuits, and to understand the activity differences between females and males. One of the benefits of this technique is to identify the targets that we can follow up later on to see the activity in these regions. And then we can take our toolkit of in vivo recordings and imaging to look at the differences in these regions. And we can correlate these deficits with behavior, and ultimately aim is to manipulate the activity of these circuits to mitigate the effects of isolation on behavior. So hopefully I'm on time with everything that happened, but so I would like to just first of all thank my lab team. Those who are particularly involved in the study are my PhD student, Matt Dawson, my technicians, Nada Jamani and Miyoto Sutsu, who are really excellent surgeons. And I'm very appreciative of our collaborators, Gina Leininger and Jonathan Epp, especially Dylan Terstiger is a talented PhD student from the App Lab who helps us with writing the codes for the scripts that we use for analysis of fiber photometry. And we're thankful for all our funding agencies, and I would like to thank you all for being here and listening to me today.
Hocam sunumunuz için çok teşekkür ederiz. Dilerseniz soru cevap kısmına geçebiliriz hocam.
Ben teşekkür ederim.
Başlıyorum hocam ilk sorumuz uygunsa.
Tabii.
Sosyal eksikliklerle ilişkili ve kadınlara kıyasla erkeklerde daha yaygın olan bozukluklarda farklı hipokretin projeksiyon nöron popülasyonlarının etkisi olabilir mi? Nöropsikiyatrik bozuklukları hedef alan yeni tedavi stratejileri için hedeflenebilir mi?
Ne kadar güzel bir soru. Bu arada soruları nerede görebiliyorum? Comments'te
Hocam
mi?
bunları ben okuyorum. Bunlar ekibimizin hazırladığı sorular. Aynı zamanda YouTube katılımcılarından da sorular geldiği zaman ekranda gözükecektir hocam. Sunumunuzu kapatırsanız gözüküyor.
Gördüm. Tamam. Evet, bu çok güzel bir soru. Evet, bu kesinlikle izlemek istediğimiz yol. Mesela otizm türü hastalıklar değil mi? Erkeklerde daha fazla görülüyor. Bunun nedeni ne olabilir? Bunun nedeni orexin olabilir mi ya da orexini etkilediği brain-wide projections olabilir mi? Olabilir diye düşünüyoruz. Bu yüzden sosyal izolasyon dışında başka bakmak istediğimiz modellerde evet otizm modelleri aynı zamanda. Ama şu anda henüz bakmadık.
Teşekkür ederim hocam cevabınız için. Bir sonraki sorumuza geçiyorum hocam. 5-HT terminalleri ve dorsal hipokampüs arasındaki etkileşimin altında yatan spesifik reseptör mekanizmaları bilinmemektedir. Buna rağmen çalışmalar anksiyetenin düzenlenmesinin altında yatan reseptör alt tiplerini tanımlayabilir mi?
Dorsal hipokampus mu dediniz? Soru öyle çıktı.
Evet hocam, doğrudur.
Evet, serotonin ve dorsal hipokampus arasında etkileşim altında yatan spesifik reseptör mekanizmaları. Evet, dorsal hipokampus aslında çok önemli. Çünkü hipokampus social memory açısından çok önemli bir brain region. Kusura bakmayın, ben böyle İngilizce terimleri araya sokarak gidiyorum. Biz daha önce başka bir grupla yaptığımız kolaborasyon çalışmasında serotonin terminalleri ve hipokampus arasındaki etkileşime anksiyete açısından ve memory açısından baktık. Social interaction açısından henüz bakmadık ama çok önemli bir konu. Şöyle bir durum var. Şimdi dorsal hipokampus, biz genelde lateral hypothalamus yani oryxin çalışırken dorsal rafedeki serotonin hücrelerini hedeflemekteyiz. Ama serotonin hücrelerin bir kısmı median rafede, yani median rafe, dorsal rafe ventral bir region, bir kısmı orada da bulunmakta. Şimdi hipokampus daha çok median rafedeki hücrelerden projection alıyor. Yani input genelde median rafeden geliyor. Yani öyle bir değişiklik var. Bu circuit'lara da daha henüz bakmadık o yüzden social interaction açısından. Ama ileride evet yapabileceğimiz başka bir şey de bu. Hipokampus social memory için önemli olduğundan hypocretin ve hipokampus ilişkisine de bakmak istiyoruz. Bu da şu anda planladığımız deneylerden biri.
Teşekkür ederim hocam yanıtınız için. Bir sonraki sorumuza geçiyorum hocam. Akut intranazal oksitosin tedavilerinin bazı hastalarda sosyal bilişi arttırdığı gösterilmiştir. Oksitosin, otizm spektrum bozuklukları ile ilişkili sosyal eksiklikler için potansiyel bir tedavi olabilir mi?
Evet. Özellikle bu şu anda klinik çalışmalarda bakılan bir konu. Ama sizin de dediğiniz gibi intranazal oksitosin bazı hastalarda açıkçası fare çalışmalarında bayağı hala devam eden fare çalışmalarında çok başarılı sonuçlar var. Bazen insanlarda tabii ki hastalıklar çok kompleks olduğu için her insanda denediğimiz tedaviler başarılı olmayabiliyor. Şu anda intranazal oksitosin insanlarda deniyor ama majority bir popülasyon buna responsive değil. Nedenini henüz bilemiyoruz tabii ki. Çok şey olabilir. İnsanlardaki hastalık, otizm çok kompleks bir hastalık. Ama oksitosin social interaction için çok önemli. Social davranış için, sosyal davranış için. Ve bizim de oksitosin nöronları özellikle paraventricular nucleus dediğimiz hipotalamus'un başka bir subregion'ı. Bizim de şu anda çalıştığımız lateral hypothalamus ile direkt ilişkide olan bir region. Bu da açıkçası bakmak istediğimiz yerlerden biri. Büyük ihtimalle kolaborasyon olarak. Çünkü oksitosin çalışan bir sürü çok yakın arkadaşlarımız var. Onlarla kolaborasyon içerisinde.
Teşekkür ederim hocam yanıtınız için. Hocam bir sonraki sorumuz YouTube katılımcımızdan geliyor. Hocam sunumunuz için teşekkürler. Çalışmanızda orexin aktivitesinin inhibisyonu dişiler hariç erkek farelerde sosyal etkileşimi bozmuş. Bunun nedeni seks hormonları ile ilgili olabilir mi?
Evet, şimdi biz de bunun nedenini çok merak ediyoruz açıkçası. Seks hormonları bizim de reviewerlardan aldığımız sorulardan biri açıkçası. Seks hormonları olabilir. Yani olamaz diye bir şey diyemiyoruz tabii ki. Yani eğer seks hormonlarıyla ilgili bir değişiklik yaparsak sosyal davranışın değiştiğini görüyoruz. Ama bunu sel spesifik olarak nasıl bakabiliriz? İşte orexin nöronlarında östrojen, androjen reseptörler var. Bunları belki aktivasyonu değiştirirsek nasıl bir sonuç elde edebiliriz? Bu bakılması gereken mekanizmalardan biri. Biz daha çok şu anda, yani bu kesinlikle çok önemli bir konu. Biz daha çok yani bilinmeyen bir konu. Östrojen ve bu reseptörler orexin nöronlarında ekspres ediliyor ama çok fazla oranda değil. O yüzden biz daha çok circuit'lara şu anda odamızı yöneltmiş bulunmaktayız. Biz bunu mekanizmaların daha çok circuit aşamasında bakmak istiyoruz. Tabii bu da bayağı uzun sürebilir ama buraya doğru gidiyor çalışmamız. Ve umarım ileride tekrar eğer size burada present etme şansım olursa bu çalışmaları gösterebilirim.
Umarım hocam, çok isteriz. Çok teşekkürler. Bir sonraki sorumuza geçiyorum hocam. How far have we been able to elucidate the basic principle that single brain regions coordinate multiple social behaviors and that multiple brain regions interact to regulate a single social behavior?
Evet, şimdi social behavior o kadar bir kere evolutionarily conserved, değil mi? Yani hepimize görülen ve önemli olan bir davranış. Ve bir sürü beyin region'ı, beynimizin bir sürü kısmı bu social behavior'da, social behavior regulation'da involve etmiş durumda. Sonra İngilizce gelince benim Türkçe iyicene. Yani bir tek hypothalamus değil. Medial prefrontal cortex var, dorsal gösterdiğim var, amigdala var, BNST. Yani bir sürü yer social behavior modülasyon ediyor. Soru şu, yani bu circuit'lar nasıl hep beraber, hep birlikte nasıl modülasyonu, social behavior modülasyonunu sağlıyor? Ve social behavior'ın da çok değişik şeyleri var, değil mi? Mesela biz onlara da bakmaya çalışıyoruz. Machine learning based dediğim şekilde. Mesela approach, ne bileyim anogenital sniffing var, işte head torso sniffing var. Bunlara da ayrı ayrı bakmaya çalışıyoruz. Bunlar çeşitli circuit'larla mı regüle ediliyor? Yani değişik social behavior'ın değişik kısımları değişik brain circuit'lar tarafından regüle ediliyor mu? Bunlara bakmaya çalışıyoruz. Ama daha çok ilgilendiğimiz bu izolasyon ya da ne bileyim social deficit olduğu durumlarda hangi circuit'lar daha çok etkileniyor? Bunu çıkarmaya çalışıyoruz. Yani bunlar bilinen şeyler değil. Çünkü multi-fiber recording yaparsınız. Yani beynin çeşitli bölümlerine diyelim aynı anda aktivitenize baktığınızda coordinated social interactions'da coordinated aktivite görebilirsiniz. Ama bu specific circuit'lar nasıl bu behavior, nasıl bu davranışı etkiliyor? Biz şu anda onu görmeye çalışıyoruz ve bu evet henüz bildiğimiz bir şey değil ama bu konuda çalışmaya devam ediyoruz.
Teşekkür ederim hocam.
Lütfen tekrardan yöneltin sorunuzu eğer.
Tabii ki hocam. Katılımcılarımız yeniden iletebilir öyle bir durum olduğunda. Bir sonraki sorumuza geçiyorum hocam. How can standardization of experimental condition be achieved for various behavior paradigms optimized in neural circuit studies?
Evet. Çok önemli. Standardization of experimental conditions. Çünkü bizim yaptığımız şu anda hani tabii ki fare deneyleri bize çok fazla çok iyi mekanizmayı çok iyi anlayabiliyoruz fare deneyleriyle ama belli behavior'lar, belli paradigm'ler optimize olmuş değil mi? Lab'dan lab'a değişebiliyor. Biz genelde yani kendi yaptıklarım adına konuşayım. Çok bilinen belirli davranış deneylerini kullanmaktayız şu anda. Bilinmeyen aktivite, beyin aktivitelerine baktığımız zaman. Yani resident intruder test. Bu kullandığımız test veya three chamber. Bunlar yıllardır kullanılan şeyler. Bunlarla başlamak istedik. Yani çok standardize olmuş bu testlerle başlamak istedik. Ama başka deneyler de yapılabilir. Tabii ki gördüğümüz çeşitli deneyler var. Biz bu territorial yani bir anksiyenin de ulaşabileceği home cage environment'ı kullandık. Bunun dışında işte fareler yeni bir context environment'a giderse behavior ve aktivite nasıl değişiyor? Buna aynı zamanda baktık. De yine çok kullanılan three chamber işte direkt sosyal davranışın yapılamadığı ama daha çok indirekt davranışların gözlemlediği testi de kullandık. Yani bu şekilde bu çalışmalarda çok kullanılan testleri yaptık şu anda. O yüzden standart olduğunu düşünüyoruz. Ama dediğiniz gibi bu da çok önemli bir konu tabii ki.
Teşekkür ederim hocam yanıtınız için. Bir sonraki sorumuz YouTube'dan geliyor hocam. Hocam öncelikle sunumunuz için teşekkür ederiz. Çalışmanız uzay ortamındaki izolasyona bağlı nörolojik bozukluklar için bir ışık tutabilir mi? Hipokretin ve oraksin mekanizmaları, öte gezegen araştırmaları ve kolonileşme bağlamında nasıl bir fayda sağlayabilir?
Şimdi soruyu tam olarak anlayabildim mi bilemeyeceğim ama çünkü uzay ortamındaki izolasyona bağlı nörolojik bozukluklar konusunda hani şöyle diyeyim uzman değilim açıkçası. Başka hani iş arkadaşlarım var bu uzaya giden astronotlar üzerine çalışan. Genelde memory'nin değişimi üzerine çalışıyorlar, hafızanın. Oradaki izolasyon sırasında neler olabileceğini, nasıl bozukluklar olabileceğini soruyorsunuz zannedersem. Bilmiyorum böyle çalışmalar var mı acaba? Çok ilginç. Ama buradan sonra gidip bakabilirim neler yaptıklarını görmek için.
Teşekkürler hocam. Hocam bir sonraki sorumuza geçiyorum o zaman. Medyal amigdaladaki nöronların moleküler olarak çeşitli alt popülasyonları, değişen girdi çıktı, devreleri ve davranışsal işlevlerle nasıl ilişkili olabilir?
Evet, olabilir. Olabilir. Şöyle diyeyim. Amigdalada evet bir sürü nöron popülasyonları var. Değişik kısımlarında değil mi? Central amigdala var, basolateral amigdala var. Giriş çıkışlar çok farklı. Aynı zamanda amigdala ve bizim baktığımız lateral hipotalamus arasında direkt girdiler çıktılar var değil mi? Inputs, outputs. Konuşmamın başında gösterdiğim insanlarda yapılan mesela amigdaladan toplanmış olan hypocretin activity, hypocretin peptides. O yüzden amigdalada evet bakmak istediğimiz bir region yani circuit açısından. Amigdala mesela fear memory açısından çok önemli bir beyin bölümü. Social isolation'da eğer bir fear etkisi varsa amigdalaya bakmamız konusunda yani bu circuit'ları özellikle yaptığımız brain-wide activity çalışmasında amigdalada bakacağımız bölümlerden biri.
Hocam çok teşekkür ederim yanıtınız için. Bu çok sorumuzdu. Aslında iletmek istediğimiz daha çok sorumuz var ama zaman nedeniyle maalesef iletemedik. Cevaplarınız için çok teşekkür ederim.
Lütfen bana eğer sormak istediğiniz başka soru varsa e-mail ile de ulaşabilirsiniz her zaman.
Teşekkürler hocam. Çok sağ olun. Değerli vaktinizi ayırdığınız, bilgilerinizi bize aktardığınız ve bilgi dolu bu konuşma için kendim ve tüm katılımcılarımız adına çok teşekkür ediyorum hocam.
Ben çok teşekkür ederim.
Çok sağ olun hocam. Çok memnun oldum. Sağlıklı günler diliyorum.
Ayrıca teşekkür ederim.
Çok sağ olun hocam. Teşekkürler.